welcome everyone to this session. In my current role, I manage effort of several trials with phase 1, and I taught several courses on drug development, including placing some emphasis on phase 1 clinical trials at Boston University. I have served as co-investigator and as a manager on several phase 1 clinical trials, and I would like to share some regulatory perspective with you and as well as practical considerations for phase 1 clinical trials. I would like to begin with a brief overview of drug development process because I realize that people come from different walks of life, and some preceding elements to phase 1 clinical trials still would be very important and very useful to be reviewed. In the drug development process, discovery is very crucial to determine clinical candidates for further testing and cell-based in vitro assays, as well as animal testing that would precede to the first exposure in humans. Animal testing is very crucial and vital to establish some efficacy parameters, toxicity, pharmacodynamics, and pharmacokinetics, and look at metabolic profile of the drug and extrapolate it in decision-making to proceed into humans. Before that happens, we need to obtain a regulatory approval of a regulatory agency in the U.S., which is Federal Food and Drug Administration, and in Europe, it's European Medical Agency, and in other parts of the world, there is a counterpart for these agencies. So this document is called Investigational New Drug Application, or IND. IND is a summary of all discovery and all preclinical work which is done before we can ask permission of regulators to transition into first-in-human clinical trials. And we supplement animal and cell-based testing with our rationale for first-in-human clinical trials. So as you can see in this slide, this is the point, a vital point of that bed to bench side first step. Transition, or as it also known as a translational research component, when we actually translate the knowledge we obtained on preclinical studies on cell assays and animal-based assays into first-in-human exposure. So the human testing actually requires at least 3 phases, phase 1, phase 2, and phase 3. They vary in terms of their objectives. They vary in terms of amount of participants that would be exposed and the goals and objectives. And in the meantime, while we conduct human clinical testing, we do not abandon our animal models, because before we transition to the phase 1 clinical trials, we have some of their acute and chronic toxicities, but we do not have long-term any toxicity data yet. That's why animal studies are still done in parallel with human clinical testing, and we always have to have plus 3 months animal data as compared to what our clinical phase would be. For example, if phase 1 clinical trial is meant to be in duration for 3 months, we must have long-term animal toxicology testing of plus 3 months, which is cumulatively must be 6 months of animal data available on chronic long-term toxicology exposure of the compound before we can expose human subjects for 3 months. So that plus 3 months rule is very important. Then when we complete our human testing, we will submit new drug application for the chemical compound or biologic license application, BLA, for biological component, which basically will have a summary, an entire life story of the drug, including discovery, animal testing, and summaries and detailed reports for the human clinical testing of all 3 phases as part of that pre-market application. And if we obtain regulatory approval, the drug or the new biological compound compound would be launched into the market. And of course, in the meantime, as we expose more subjects and as we prepare to put it on the market, we scale up our manufacturing. So there are some ongoing regulatory obligations throughout the life cycle of development of chemical compound to biologic. We will emphasize transitions. We will talk about some specific regulatory requirements as it pertains to phase 1 or first-in-human clinical trials, on which we will focus this session. And as you can note, that their main focus here is safety, safe transition from animal models into human subjects. So that's the key focus of phase 1.